Hi everybody, this is Kostas Konstantinidis. I'm one of the co-organizers of this workshop and also a professor at Georgia Tech. I'm going to give you today my perspective about whether or not metagenome assembled genomes or MAGs and single cell amplified genomes or SAGs can be of the same quality as isolate genome for taxonomic descriptions. The issue, to give a summary, is that in order to describe a new species or a new higher rank, you need type material. You need like a voucher. And currently only pure isolates are allowed as type material. A genome sequence is not appropriate for this. Actually, the proposal to use a genome sequence as the voucher was voted down recently, like last year. And the main reasons I heard in the literature about this are summarized here. So basically, it has been argued that the genome sequence is not appropriate because it doesn't represent a living organism, a living fossil. And specifically for the MAGs, it doesn't even represent an actual cell, an actual organism, but it's rather the composite genome of many isolates. These are valid points that are true. I agree with them. However, what I want to say is I don't think we need a living organism in order to describe the diversity patterns we see around us in nature. Like, what is the species, the unit. We don't need to have the organism living somewhere in our labs. Other taxonomies, non-prokaryotic, they don't require a fossil fuel for this purpose either. And for the MAGs, I will argue that having a composite genome is actually an advantage compared to having a single organism, a single cell genome, because sometimes this single cell could be an outlier compared to the total population. It has been argued also that the genome doesn't describe the relevant phenotype. I think because the genome shows the potential phenotype, but it doesn't mean that the phenotype is expressed. So to express it, you need to grow the organism at the appropriate conditions. And again, I think this is a valid point. However, what I will argue is the genome is the starting point. It helps you make hypotheses about what the phenotype will be. And also, perhaps more importantly, the way we assess phenotype using some of these traditional taxonomic methods, like for example biologs, or biolog plates, or lipid profiles, or growing even the organisms in media that are very artificial compared to the natural conditions, and they are artificial. I will also argue that it gives us irrelevant phenotypes also. So again, I think it's a valid point, but not a big one to prevent progress and using genomes as the type. Along this line, it has been argued that it's going to put the clinical macrobiology at risk. I think the argument is again similar, that you may have a genome, but you don't know if it's going to be expressing virulence, it's going to be a virulent pathogen. And I think it is also a valid point, but almost all pathogens of humans and many animals are known. So we know what are the virulent genes, even sometimes what mutations they have that prevent them from expressing. And so having a genome is not going to compromise that. It's going to actually tell us, okay, this has the potential to be a virulent organism. So I don't think it's a big concern either. For novel pathogens that haven't been described, the genome can be the starting point of making hypotheses, as I said before. So again, I don't see this as a big concern to prevent progress with respect to type material. Finally, it has been argued that the MAGs are often of insufficient quality, and also when sequencing technologies change or the bioinformatic methods change, you're going to have new versions of that genome, and so with new versions comes instability because it's not going to be exactly as the one before. And the instability applies also to the genome of isolates. My argument here again is a valid point, but if we go upfront for high standards for MAGs, and I think the technologies are mature enough right now, the new versions are going to change really very small things, like some SNPs here and there that were called wrong, but I don't think it's going to change the gene content. It's unlikely to change the gene content, etc. So even though, again, these are valid points, the difference will be very small. It's not going to change the phylogeny of the MAG or the potential phenotype it has. So overall, my view is that if we go for high standards, we are going to have a stable system, stable enough system. And I'm going to make the argument by showing you an example that I don't think using the genome as the type material will take away focus from culturing the organisms. I actually think it's going to make it more intense. And what I'm going to do next, I gave you the summary and my view. I'm going to do next an example to basically back up what I just said. So my example comes from the Gulf of Mexico, the oil spill there. So myself and collaborators have been studying the BP accident that happened in the Gulf of Mexico 10 years ago now. And basically we have been to the beach and collecting samples where the beach was very contaminated with oil. You see some pictures here in the middle. Some of the analysis we have done and some of the most interesting data. So we did applicant sequencing of samples before the spill, here on the left, during the spill, and then after the spill when the beach was clean. And what really stand out in this work was there was a very abundant 16S OTU. You see it here with a red color, making up 20% or more sometimes of the total community that stand out. And you know, in natural communities like beach sand, this is very unusually high abundance. You never see something so abundant. So we were very curious to know what this organism is, and so we took metagenomes from these samples using some of the established tools for these purposes for assembly and genome binning, and also as well as some scripts that we have made available actually publicly through this publication on the web server. We managed to get the draft genome of MAG01, that's how we call it, of one organism that we think it is this red guy that I just showed you, based on its abundant patterns in the metagenome. So it was also very abundant in the metagenomes. So we were very curious to know if this is a known organism or it is a novel one. So we then turned to the Microbial Genome Atlas. Microbial Genome Atlas, or MIGA for shortcut, is a collaborative effort between my group and Luis Miguel Rodriguez, who is now an assistant professor at the University of Innsbruck in Austria, and also Jim Tigi and Jim Cole at Michigan State University. And so what MIGA basically is, we have collected all the genome sequences that have been classified, so we know what species they belong to, and have made them this a searchable database. So somebody, a user, can query an unknown genome against the MIGA and figure out if the genome is a member of an existing species or if it is a novel species, and MIGA can actually tell if what is the level of novelty. Is it a novel genus, a novel family, and so on. MIGA is using the genome average nucleotide identity, and ANI, and the genome average amino acid identity, AAI, to do that. So, you know, check it out. Here is the website. When we searched this MAG01 against MIGA, what we saw was there was no match, close match at the species level or even the genus and the family. So our organism is probably a new family, because the closest match was a genome of the Chromatiales order of the gamma proteobacteria, and the AAI was very low, in the around 50. So we were sure we have a novel organism, but we didn't know if it is novel, for example, against organisms that have been only named by 16S. You know, some of the organisms are still don't have genome sequence and they are only known by the 16S. This won't be in MIGA. And so the MAG we got from the environment didn't have its 16S, as it is common for this complex environment like soils, beach sands, even the ocean and freshwater system. So we didn't have the 16S to check, but what we have in one of the contigs at the very end, there was a piece of 16S. And so basically with PCR walking, having a primer sitting on one specific area of our contig and the universal primer for the 16S for the reverse, we did PCR walking and we were able to link our MAG to a 16S. And then we confirmed that the 16S is also very new. But not only that, because we had the 16S, we went around and looked for applicant data from oil environments, and we have shown that this organism is very abundant wherever there is oil, making 10–20%, even in Antarctica spill oils, as long as it's a coastal sediment. So this work was published. This work was published in ISME, and we were quite surprised to find such a dominant organism that hasn't been described before for such a key process like oil bioremediation and oil spills. Of course, the next step for us is to grow it, because really, if you have the organism in culture, you can start doing some nice experiment. You can use it as a bioremediation agent. You can study more the success of it. So our next step was to look at the genomes and try to figure out why it's so successful, how we can grow it. And so we did some bioinformatic metabolic modeling based on the genome, and we saw that it has the genes for alkane degradation, as we were expecting, but also it has some other genes that probably help it to be so successful in oil, such as it's a nitrogen fixer, oil is usually nitrogen limited, and a few other very interesting observations from the genome. So using this information, we then made some media, hopefully to get this organism in pure culture. And the media were, we gave hexadecane as the carbon source and only carbosomes. We didn't put any nitrogen because we know this organism is a nitrogen fixer. And we used the 16S in a colony PCR in order for all the organisms that grow on our plates to figure out which one is this the right one. So after about 2 or 3 months, we were successful, and that's the picture here. You see, it's a real picture. So the red thing, this one, is an oil droplet, and you see on it several cells of this organism, or on the side, like here, to grow. And so basically, we were able to get it in pure culture. We named this organism Candidatus macondimonas deazotrophica. And this was published, as I said, last year in ISME. This was a case of success, and we sequenced the genome of the isolate, and basically we found that it is exactly the same as the MAG. So it has 99.9% ANI to the MAG, no gene-coded difference, and it has the same genes for nitrogen fixation and alkane degradation. In addition, since we had the isolate, we have now determined, what is the range of tolerance in temperature, pH, and salinity, and we are in the process of depositing to culture collection, so we can lift the Candidatus and have just Macondimonas deazotrophica for its name. So what I hope with this example I show you is you can do it. The MAG can be a great starting point. You can and further promote isolation, because at the end of the day, if you have the organism in hand, you can do so much more, and we are working on this organism much more. So to finish with some conclusions, I think the MAGs and SAGs can be of good quality, similar to the culture descriptions, to the genomes of cultures. They are not perfect. There are mistakes sometimes, but we have now the means, the bioinformatic means to detect these mistakes. And I will even argue that methods that we are using taxonomic were never perfect. So for example, this is a picture from one of our papers that did the comparison of the DNA hybridization to ANI. That's where the ANI, the 95 cutoff, came out actually. And so every point is a comparison between two genomes, and you see the DNA hybridization versus the ANI value for these genomes. And you see that there is quite a bit of variation from the mean, and I'm pretty sure this is because of the experimental noise of this method as opposed to the ANI. So what I'm trying to say is that the methods, including the DNA hybridization, is not perfect. It has noise, but this hasn't prevented us from making progress. And my view is that the MAGs and the SAGs are even better with that respect, although they are not perfect, as I said. And so I think we can move forward. I think the methods are stable enough, and importantly, they are accessible to everybody. And I hope with the Macondimonas example, I convince you that it's not this change in type material is not gonna take focus away from culturing. The standards, myself, Ramon Rosello Mora, and Rudy Aman proposed some standards in 2015. So for example, to have MAGs of high completeness and low contamination, to try to get the RNA gene, the 16S, because there is so much more you can do with it. And in addition, to describe somehow the phenotype, at least the potential phenotype bioinformatically, if you can prove some of these functions experimentally with some culture-independent methods, that would be really cool. But as a minimum, you should have some prediction from the phenotype. And when you meet these standards, I think you should be able to describe a stable enough species, etc. If these standards are not met, then I don't think there is a need to describe these organisms. This can stay as an accession number until somebody is more interested in them and they do the extra step. So I think, these standards will change as technologies change, but I think it's a good starting point for us for discussions, etc. And along this line of, changing standards, etc., very recently we published a paper where we compare mugs with isolates coming from the same sample. And basically we confirm that these standards are really good, but in addition, we're going to propose that if you have mugs or sags to have at least 10x coverage by reads, and also check potential chimerism with read recruitment plots, because sometimes even if you have these high standards, because they are based on only the universal genes, not the whole genome, you can still have very chimeric mugs, and this paper describes an approach to correct for these, detect and correct these issues. So this is not any more sufficient in my view, but it would be nice to include some of the extra steps I have here. That's the end. Here is the website where we have some of the tools that I mentioned, and thanks to my our funding agents for enable this work. And thank you very much for your attention.